right. Welcome everybody to our newest NIRx webinar. I want to offer a very warm welcome to Dr. Jiaming Zhao, who is going to be speaking about the spatio-temporal tomography of the brain, the DOT-EEG fusion algorithm, and the new NIRFASTer. Before we get started though, I just want to give a short introduction as to who we are. We are NIRx, and for approximately the last 20 years, we have been providing researchers such as yourself with innovative systems for your research, predominantly involving near-infrared spectroscopy, FNIRS. In addition to the wonderful systems we provide, though, we want to highlight our support system. We strongly believe that it's not useful for us to only provide you with great systems if you don't know how to use them. And so we want to support you throughout the entire process from your beginning of your research all the way through the end so that you can not only collect great data, but publish those papers, get those grants, and really not only move forward the science in your area, but also the science of FNIRS in general. So if you ever have any questions, concerns, or difficulties that arise in your research, please don't hesitate to reach out to us. We would be more than happy to help you. It's really what we love to do. If you would like to get in contact with us, you can connect with us at any of these social media platforms. And again, like I mentioned, support is our really big emphasis in addition to our systems. And so if you have any questions, comments, or concerns, please don't hesitate to reach out to us at thatconsulting@nirx.net, and we'd be happy to help you again. Additionally, if you would like to subscribe to our newsletter, we promise we won't spam you weekly. We aren't going to overrun your email inbox, but you can use that QR code or that website to access our newsletter. About 3 to 4 times a year, we will send you an email containing the latest and greatest product features and releases. We might feature some innovative research that's going on in our communities, as well as other exciting news that might be of interest to you. Next, I just want to lay down some webinar considerations, some ground rules for you. Please note that you will be muted throughout this webinar, but if you do have any questions that pop up throughout the webinar, please don't hesitate to use that Zoom chat to submit questions, and we will get to them at the very end during our question and answer section. Additionally, this webinar is being recorded and will be uploaded onto our website in the upcoming days. And let's say in a couple hours after the webinar is done, a question pops up that you want answered. You can always submit that to consulting or support@nirx.net, and either we will answer it or we'll submit it to Dr. Zhao, who will be able to answer that for you. Last little bit about us. My name is Amy. I'm a scientific consultant here at NIRx, and my colleague Cami, who is our marketing and events coordinator, we will be moderating this webinar. And now to the real reason why we're all here, I would like to introduce Dr. Jiaming Zhao, who is currently a research fellow at the University of Birmingham. His research right now is predominantly focusing on advancing algorithms for high-speed, high-accuracy diffuse optical tomography. And with that, I want to wish you all happy learning, happy webinar, and hand it over to Dr. Zhao.
All right. Hope everybody can see my screen. Good. Thank you very much for having me here. Thank you very much for the introduction. So let me get started with the presentation if that's the case. Right. So the title is Spatial-temporal tomography of the brain, the DOT-EEG fusion algorithm and new NIRFASTer. As the title suggests, there's going to be 2 parts of it. And if there's no objections, I'm going to start with the first part first. That is the neuronal source reconstruction using joints, DOT and EEG. So I think most of us should be familiar with the EEG and DOT, but just in case, I'm going to very quickly go through what these things are and then why we care about the fusion of them. So EEG is basically a recording of the electrical activities in the brain. Oops. There we go. Or sometimes we call them neuronal sources. And on the scalp, non-invasively we can record the electrical recordings through the electrodes. And so there's a little illustration of how the system works. And of course, based on the scalp recordings, we can also make inferences on the sources that are inside of the brain. And this is something we call source localization or neuronal source reconstruction. And DOT is really what NIRFASTer shines, right? So basically the idea is that when the hemodynamic changes happens in the brain, specifically we have two species of hemoglobins that we typically care about, and that will lead to optical property changes of the brain. And if we measure with light, that will cause light intensity changes. And that's the one sentence physics behind the idea of FNIRS or DOT. And of course, we can do the same thing. We can back propagate from the light changes that we record from the scalp and make inferences on eventually the oxy and deoxyhemoglobin changes that are inside of the brain. And you might be familiar with FNIRS, but not so much DOT. So here is the only difference, which is that if you measure these bananas at many different locations that are overlapping, which would be the best case, then you can have a 3D reconstruction of this anomaly that is inside of the brain. So that is the only difference between FNIRS and DOT. And of course, you might already know that the two different species of hemoglobin would have different absorption spectra, which means that if we can measure with two wavelengths or maybe more, we can separate the influences of the two different species of hemoglobins. So that is a very brief introduction of EEG and DOT. So hopefully we're on the same page now. So here's the idea of combining these two things. So I was doing some research in the lab, and I was dealing with both of the two different modalities. And the observation is that, well, I guess it is not so much an observation of myself. It's a known fact that EEG is temporally very fast, but if you do this source localization, the point spread is typically quite large. So it looks something like this. It's pretty blurry, even though it should be just a point. DOT, on the other hand, it can be spatially very accurate, especially if you use a high density system and then processed appropriately. But the issue is that it measures hemodynamics that is inherently slow. So we can use devices that sample at fairly high sampling rate, but that's not going to help because that's the nature of the physiology that it measures. And now the idea is there a way to combine these two different modalities? So one is fast, one is spatially accurate. So, we can have the best of both worlds and have a spatial-temporal reconstruction of the neuronal sources. So that is the idea. And you may still not be very convinced, but of course we can do that. But why do we care? So what would be the applications cases? Well, we can say that we care about the spatial-temporal dynamics that is happening in the brain, and then using either one of the modalities would not suffice. For example, if we want to decipher how or to monitor how the information flows in the brain, so you see something and you want to figure out how the information flows from the visual cortex all the way to the higher level cortices of the brain. Or, for example, if we want to localize the focus of the epilepsy, but non-invasively, or we care about the neuroscientific side of functional activity, let's say how it evolves with time, what are the temporal changes of functional connectivity, and then many others. So that is the basic idea, and also the motivation or potential applications behind it. And if you have a background in fMRI, you might be familiar with the similar kind of work that is already done in EEG and fMRI fusions. In that case, that would be EEG would be providing the temporal information, and fMRI would be providing the spatial information. And we combine these two things, we have this spatial temporal reconstruction of neuronal sources that typically using fMRI as the spatial prior to constraints the EEG reconstruction part. So that is already done. But what we are trying to do is to replace the fMRI with DOT. Of course, fMRI is still the golden standard, is a great modality to use, but of course, it is not free lunch. It is quite expensive, and it is quite bulky machine, and is not compatible with people with metal implants. It is not bedside compatible, and so on and so forth. So at least in certain circumstances, fMRI is not necessarily the most preferred modality to use. And DOT, of course, the fundamental limitation is that it measures quite shallow. It's only a few centimeters deep. It's very difficult to measure the subcortical structures. So that we know, but it does have this very large advantage of being portable. For example, this is a picture that I took from the NIRx website. You can basically be wireless. You can put this device in this vest that comes with the device, and you can do something quite organic and still be recorded without making too much of a mess. So that is the big advantage. So that is why we want to substitute fMRI using DOT and see if we can still do this kind of spatial reconstruction using DOT and EEG fusion. So that is the motivation, and that is the one slide summary of how that should work. Now we can start to dive into the details for a little bit. So I mentioned that I would like to use DOT as the spatial prior to constrain the EEG reconstruction, but how exactly is that done? All right, so suppose we have this voxelized space. The brain is basically discretized in space, and the brain would be represented in different voxels, which is basically a 3D version of a pixel. Suppose we take one of the voxels and we measure its hemodynamics, or rather reconstructed hemodynamics. That is, of course, done using DOT. And we make this assumption that if the hemodynamics has a strong amplitude, a strong activation, we say that we assume that the neuronal activity would also be strong, which would be reconstructed using EEG. And vice versa, if for this particular voxel, the hemodynamic has a low amplitude, then we also assume that the same voxel will have a low amplitude in electrical or neuronal activity as well. So this is how we relate to these two different modalities. And if you are familiar with physiology, you might have already noticed that, okay, we're making a fairly strong assumption on intact neurovascular coupling. And yes, that is true. For those who are not familiar with what neurovascular coupling is, here is, again, a one slide summary of that. So, well, there are different variants of how you should define or view this neurovascular coupling phenomenon or mechanism. But here's one version of it that is, I suppose, the most simple. So basically, you have some neuronal activation, and you have some electrophysiology changes, that is due to the ion flows in the neurons, and then locally, and we because of some sort of chemicals and other mechanisms, that causes the hemodynamic changes. And the relationship between the electrophysiology and the elicited hemodynamics would typically be modeled by this hemodynamic response function. We call it HRF, that usually takes this kind of shape. So yes, that is an underlying assumption. And from the first two slides, we know that the electrophysiology is picked up by EEG and hemodynamics is picked up by DOT. And because they come from the very same source, at least that is the assumption, we can combine these two pieces of information and have a better inference of the underlying neuronal activation. And of course, the concern is that what if this link between electrophysiology and hemodynamic is broken, or the shape of the HRF is not what we assume it to be? Then yes, that would be the limitation of this algorithm that I'm going to present. Although I would say that in most cases, especially under controlled experiments, this in general holds quite well, and that is of course supported by a lot of fMRI literature and others. Another thing is that it will become clear later in the presentation. Should this HRF change its shape, what we can do is that we can in the first step, quantify the shape of this HRF and substitute that into the algorithm, and then theoretically that should still make the result hold. So that was a very long one-slide summary. Now let's dive into this reconstruction problem. So I would like to, because we do have quite a bit of time, so I would like to discuss some of the mathematical details of this algorithm, and hopefully that would be of interest of some of the audience, if not the majority. So suppose we have this linearized measurement problem, which is in general true for both EEG and DOT. So recording, Y is the recording vector, which can be either EEG or DOT, it doesn't really matter, or something else. And A is the forward matrix that is modeled based on the head shape and the physics of the problem itself. And beta would be the brain activities. In EEG, in the case of EEG, that would be the electrophysiology changes. And in the case of DOT, that would be the optical properties or hemodynamic hemoglobin changes. And epsilon is the recording noise. And what reconstruction does is that we have this scalp recording of Y, and we're going to make inferences on this beta vector. And this is typically done using something called Tikhonov regularization in DOT, or the same thing is called M&E. This is in the world of EEG, that is short for minimum norm and estimation, or you can call it ridge regression, or there are different names, but essentially that's to solve this optimization problem, which has a square error term and a penalization on the energy, where lambda is basically a scalar that controls how strong this penalization is. Yeah, so regardless of the recording modality, may it be EEG or DOT, it effectively boils down to this very simple equation in at least in many applications. And here, it is necessary to use a different formulation in order to incorporate DOT prior into EEG. This is done in collaboration with Ted Hubbard at the University of Pittsburgh, who actually mentioned that this DOT-EEG fusion algorithm was done at Carnegie Mellon when I was doing my PhD. I will be mentioning the names at the very end of the presentation, but let's stay here. This slightly different, well, maybe not so slightly, but this is different. This different formulation is called restricted maximum likelihood, or REML, as you can call it, or it can also sometimes be called hierarchical Bayesian. If you have a Bayesian kind of view instead of frequencist. But it looks much longer, but if you look carefully, it still has a square loss term, an energy term, that is the penalization. And the only difference is that instead of a L2 norm, here we have this weighted norm. And the weight would be coming from the C and CP matrices, which also appear here. And that is actually the secret sauce of this different formulation, or this Reynolds method. So these are the covariance matrices. We can assume that we know these things. We can estimate them from other means and just plug it into this equation. In which case, in this optimization problem, the minimum will not be taken with regard to C and the CP, and they will disappear because they will become constant. But here, in this REML formulation, we can actually estimate these two covariance matrices from the data at the same time as this brain activity vector itself. And if, of course, this is for each time step. If we do this for the entirety of the time trace, we'll have a temporal reconstruction of the brain activities. And really, the most important is the CP matrix, which is the covariance matrix of the neurons. And this is where the DOT prior enters the EEG reconstruction. So let's have a closer look at it. So suppose we have this DOT reconstruction, and this red blob would represent something that is reconstructed as strong activity. And we mentioned that we care about this voxelized space. So if we use this voxelized space and vectorize it, so pay attention that this is not a time trace, but this is just voxel space vectorized. This bit with high activity would correspond to this little red patch. And using some sort of smoothing, we can put this vectorized version of DOT reconstruction as the diagonal of the CP matrix. So the idea is this: if one voxel has a high amplitude, then it swings larger, which means the covariance, the variance of this voxel would also be larger. And of course, if the amplitude is smaller, it swings a little bit less, then the variance of this voxel would also be smaller. And that is why we give voxels that has a strong activation, or assumed to have a strong activation, a larger variance value. Or we can represent it this way, say that suppose we have a voxel I, and if the DOT has high amplitude, the DOT reconstruction of this voxel has high amplitude, and then we assume that the diagonal, the corresponding diagonal element of the CP matrix would also be high. So that is the intuitive explanation of what I'm going to present next, or maybe you find the mathematical representative more intuitive. I don't know, but here it is. Suppose we have this DOT reconstruction of oxyhemoglobin. Let's only care about the oxyhemoglobin because it's typically more reliable in reconstructions. So we'll use that as the spatial prior. Let's call it beta hat. And we normalize it to this largest amplitude. And we take the absolute value and then normalize it to the largest of all absolute values. So this beta prime vector will be somewhere between 0 and 1. And finally, how we construct this covariance matrix CP is that for each element II would be essentially a scaled version of this corresponding element in this beta prime vector, where A and B are just hyperparameters. Of course, you can use some different formulations of how you relate this CP diagonal and beta I. This is basically to say that we want to adjust how strong the prior contributes to this reconstruction problem. And I think in EEG fMRI combination, sometimes we will use something much harsher than this. They have this 1090 thingy. That would be a hard thresholding. This is a soft thresholding that incorporates the DOT prior. And if you have been looking at this equation for a while, you might notice I'm not using an equal sign, but a proportional sign here. So what does that mean? And I do like to make a few comments on this Remmel or hierarchical Bayes method, which is that very nicely we can assume a decomposition of this covariance matrix. I'm using CP here as an example. You can use the other covariance matrix for the measurement noise as well, and then do the same trick. But basically, you can assume a decomposition. You can assume it has it's a linear combination of several components. For example, you have this spatial prior. You can have a functional connectivity that you estimate somewhere else. You can have or for example, it can be a smoothing of nearby voxels, and you can have something else. And we mentioned that this Remmel algorithm would estimate the CP matrix from the data while it runs. And if you assume this sort of decomposition, what it boils down to is that it's going to estimate this lambda factors that is in front of each of this covariance matrix component. And and of course, if you make some reasonable educated assumptions on decomposition, hopefully theoretically it's going to help you dig out some of the hidden information from the data, because you're effectively imposing some further constraints on how the algorithm should work. Of course, you have to be reasonable. You shouldn't make something that is completely irrelevant, and then the algorithm will not run very well. So that is the nice thing about Remmel or hierarchical Bayesian methods in general. And what we are doing is effectively a special case of this, where we assume that there's only one component that is related to the spatial prior. And here we see why I use this proportional sign, because this lambda one factor would be estimated by the algorithm itself, and all we're assuming is this QP that is on the right hand side of this bottom equation. Okay, I think we should move on to the results, but before doing that, let's stare at this main equation for the last time. And here we know that Y and A are the recording and forward matrices of a problem, and in this case we use the EEG version because we're using this asymmetrical, we call it asymmetrical fusion, where the base of the reconstruction is still the EEG itself, and DOT is used as a spatial prior to constrain it. So here, this is just a modified problem of the EEG reconstruction, and of course this is the EEG part, and DOT prior is incorporated through the CP matrix, as we just discussed in the previous slide. And after this combination, the result of this this optimization problem, the solution to it, the beta will be, well, the beta that optimizes this loss function would be this our desired reconstruction of DOT plus EEG, or the spatial-temporal reconstruction. And how we incorporate this DOT prior is that, again, recalling from the previous slide, for each voxel I, if the DOT is reconstructed to have strong amplitude, a high amplitude, then we assume this corresponding diagonal element of the CP matrix to be large. So here is the mathematics behind this DOT plus EEG algorithm concisely. And now it's time to move on to some results. So we started with some simulated data. Suppose we have two activation spots that are sequentially activated with a fairly short time separation. They're also spatially quite close. This is effectively saying that if you use DOT only, because they're so temporally close to each other, there's no way we can separate them, the onsets of them. And they're so spatially close that with the blurriness of EEG reconstruction, we can spatially confuse these two activation spots. And that will necessitate the combination or the fusion of those two different modalities. And hopefully that will help us spatially and temporally resolve these two different spots. And this is the simulated grid that we used. So if you look at this this image here, the black dots would be the locations of the EEG electrodes, and the red circles are where the DOT source is assumed to be. The blue circles are where the detectors are assumed to be, and A, B are the activation spots, and the green dashed lines would be the source detector pairs that are formed. So here's the measurements grid. And if we do this DOT reconstruction, if we is something like this. So it reconstructs both the spots very well. You can clearly see A and B location. Of course, the temporal information will be lost because they're so close to each other. There's no way we can resolve them. But spatially, it's very correctly resolved. And what does the spatial temporal reconstruction look like, or what does the fusion look like? Well, here's a comparison of a few different things. We'll walk you through them. The first row is the ground truth. This is what we wish to see. And I'm showing three different timestamps. Remember that we have these two bell curves. And 50 milliseconds would be where the first one peaks. 100 milliseconds is where is the spot B peaks, and 75 is about half and half. And if we use only EEG, as we expected, because of the blurriness or the large point spread of EEG reconstruction, we kind of see that two different things are happening, but if we pay attention to this 75 millisecond case, there's no way we can tell that there's two different places that are simultaneously activated. But if we apply the DOT prior, it looks much nicer. Not only that, when at 50 milliseconds and 100 milliseconds, the reconstruction is much more confined, but also when these two spots are equally activated, we can fairly clearly see that there are two different spots. If we compare that to here, where it's just a big blurred blob. So that is really promising. These are quite close to each other's spots, then there are sequential activated, and then we can do this kind of very nice reconstruction if we fuse these two different modalities. And of course, now it's time to move on to a in vivo validation of this algorithm, because if you cannot validate it in realistic data, then it's still not very convincing. So here I'm using a simplified version of a very classic retinotopy experiment. Before describing the experiment, I would like to briefly introduce how this visual cortex works. So suppose you see something in the visual field, that is this blue box on the left. And if you see, well, if I were to divide the visual field as well as the visual cortex into 4 different quadrants, it works in a mirrored fashion, where if you see something in the top half of the visual field, then you would expect some activation in the lower part of the visual cortex. And if you see something in the bottom part of the visual field, then you expect some activation in the the top half of the visual cortex. And the same mirroring effect is also applied to left and right. So that is why you see this. So this is a very classic example in high density DOT. So essentially what happened is that the subject is given this stimulus of this rotating checkerboard, and you measure from the visual cortex in the back of the head, and then you do this reconstruction. So this is the video where I think the bright yellow would correspond to this high activation. And you see that they're rotating at the same speed, but at an opposite position because of this mirroring effect. So this is the classic example experiment. Although using that full rotating checkerboard is a little bit too complicated for just a proof of concept experiment. Therefore, I used a simplified version of it, where instead of showing this a full rotation, I'm only giving the subject two different locations of the checkerboard. So one is on the left, on the bottom left, then you expect some activation on the top right of the visual cortex, and the second location is on the bottom right, and in which case you will expect some activation on the top left of the visual cortex. And if I rotate between these two things at a fairly high speed, then DOT itself will not be able to tell apart when is which happening. And EEG, of course, will be able to tell them apart, but so with the blurriness is not going to be spatially very accurate. And with the hope is that if we apply the DOT reconstruction as the spatial prior for EEG, we can not only resolve these two different places, different locations of stimuli, we can also make them spatially more confined. So that's the idea. The protocol is something like this. We start with a resting state, but then we have a stimulation period of 15 seconds, and we have this resting state of 25 seconds just to make sure the hemodynamics will regress to resting state. And within this stimulation, we have this left checkerboard for 200 milliseconds, and then it's blank screen for 175 milliseconds, and this right checkerboard, and repeat. This left-right cycle will be repeated for 20 times, and if you do the math, it's approximately 15 seconds. And this is what we call a block, and this block is repeated for 25 blocks. So each run of the experiment is approximately half an hour. So here's the experimental setup, and special thanks to Professor Barbara Shinn-Cunningham, who very kindly offered the lab space, and the personnel who helped us with the setup. So basically the subject would be seated in front of a monitor at a fixed distance. And simultaneously we measure NIRs, that is a NIRx NIRSports2 device. We're using 24 sources and 21 detectors, forming quite a lot of channels on the visual cortex. It is measured at two different wavelengths. And the EEG is a biosemi system. It's got 64 electrodes. So here's the experimental setup. And this is what the cap looks like. So this is a biosemi cap, and we installed these electrode holders and output holders ourselves at our design locations. So if you look at this picture, the black parts would be the output holders for the NIRx devices. The white bits would be the standard biosemi electrode holders. And this is what it looks like when it's populated and put on the head of a subject. So yeah, it looks quite cool. And if you look at this 2D version, these are where the source and detectors are, and so how the channels are formed. If I were to put this optical locations into a standard 1010 EEG grid. So yeah, that covers quite a bit of the brain. And I will briefly go through the data processing. On a very high level, DOT, I use this GLM model, and EEG, I use this standard trial averaging technique. And the forward models are constructed from the same template brain. We don't have the individual MRIs of the subject, so we just use a template brain. And of course, we need to fuse these two different modalities, and then we have to use the very same brain model. And if we want to know about the details, here's the details. We convert it to optical density changes, bandpass filtered, and then we have this GLM model. EEG is a very similar standard procedure where we did do this epoch averaging. So the results that we present will be on a averaged epoch that contains one left and one right checkerboard position. And this forward model is constructed from this ICBM 152 model, Brain Atlas, and then use NIRFASTer, which I will discuss later, and field trip to construct the forward models for the two different modalities, respectively. And they're reconstructed using T-clone regularization. That's the baseline reconstruction. And the baseline reconstruction, well, the classic reconstruction of DOT will be then used as the spatial prior of the EEG reconstruction, in which case we'll move on to this REML formulation or the hierarchical Bayesian. To briefly discuss this GLM design matrix, we have this DC bias, we have this linear trend, we have this something that is related to this protocol itself, that is the stimuli involved with HRF. And we have this accelerometer and gyroscope data that is both recorded by the NIRSports device. And if you recall from the beginning of the presentation, when I discussed the neurovascular coupling, saying that if the HRF changes and if the changes are quantified, we can still make the algorithm to work. That is because of this. So theoretically, if you substitute this HRF using something that I quantify from a real data, it should work even better than assuming a canonical one. And here's how you and where you can incorporate this piece of information. So enough discussion of theory. Here is a result of the DOT reconstruction. This is a group average of 16 subjects. This is something that we published last year on NeuroImage. If we look at these two checkpoint positions, then we expect some activation in this top half of the visual cortex, which are indicated by these green boxes. So the green boxes are basically to draw your visual attention. It's not scientifically calculated or anything. It's just a visual indicator. And you see this reconstructed activations at the expected locations. And of course, you can mask it using a p-value and it looks a little bit cleaner. And this p-value masked version of oxyhemoglobin reconstruction, or this panel C figure, will be used as the spatial prior for EEG reconstruction. And here is what it looks like. I will pause here for a few seconds so you can have a look at the animation. So hopefully you can see that when the stimuli are shown and changes, you will see the propagation of reconstruction that is happening on the brain cortices. And if you look at the comparison between these 11 2 animations, you will see that hopefully, when DOT prior is applied, the activation reconstruction is much more confined and spatially accurate, while dynamics is still there and preserved. And if that is hard to see, we can look at a snapshot. For convenience, if we name this left and right locations as stimulus A and B, and if we look at the strongest activation spots, which would be 170 milliseconds after the onsets of the two different stimuli, something like this. Just a little reminder, this is the prior that we used, this little figure in the middle. And so on the bottom, on the top, is the EEG reconstruction without the use of DOT prior. You do see that we see there's a reconstructed activation at desired locations, but they're quite blurry. But if we apply this DOT prior, it looks much more spatially accurate. So this is the theory and the results of this NeuroImage paper that we published last year. So we would like to call it success. This is really promising in the sense that if we fuse these two different modalities that are simultaneously recorded, we can theoretically do this kind of a spatial-temporal reconstruction of neuronal sources, and that is indeed validated using both simulated and human retinotopy data. That is very exciting, but of course, you might be thinking that this confinement in spatial content, is it real or is it potentially over-constraining? And indeed, we don't have an answer to that because we don't have the ground truth for that. Ideally, would like to have fMRI recording happening at the same time as well. And in that case, we'll be able to tell if this is over-constraining or if this is true. And of course, this is one control experiment in our lab, and that is successful, and we'd like to look into more realistic applications. And of course, another thing we can look at is to have the spatial prior even more accurate. So this is a peek into the future. We do some deep learning things. So basically, we have this reconstruction. We plugged it into a neural network, which will give us this enhanced reconstruction. There's something like this. This is what we presented at the FNIRS conference that happened this year here in Birmingham, actually. So I think I'm running a little bit out of time. So I will very quickly go through this second part, which is the new NIRFASTer. So this is the brief introduction of the history of the NIRFAST toolbox. This is something that Hamid Degani, my current advisor, did many years ago. So effectively, it's an FEM solver of the diffusion equation, and it calculates the fluence that is propagating in the turbid medium. And that's the FEM solver is the heart of it. So it started way before that, but the first monumental moment would be this 2009 paper. And the second monumental moment is back in 2018 when a postdoc here, Dr. Stanisław Wojtkiewicz, developed this GPU version that is called NIRFASTer. That is uses CUDA acceleration and parallelized algorithms, and it's faster. Here it's the bunch of things that it supports. I didn't make these figures, but if you're curious, you can go to this GitHub website and have a look yourself, because there's a lot of information. And now we're moving on to the Python version. So that is where the new version is. That is objects oriented. And according to Hamid, the name should be Fast and Furious. So the main object would be a mesh that can be loaded through different sources, where the ASCII is really the classical NIRFAST format that we use now in the MATLAB version. And we're supporting 3 different mesh types, and within each type we can generate these data and Jacobian as well. And we're using updated solvers that are even faster than what it currently is in the MATLAB version, and we also provide the analytical solutions in semi-infinite space. These are exactly the same functions as the MATLAB version, but ported to Python. So here's what it supports, and here's the link if you haven't figured out. So very quickly go through the pipeline. What do I mean by object-oriented? You can load the package, load the mesh. It's going to spit out something. And by doing this, you are automatically representing the data on a regular grid. And generating forward data is just this one line. Suppose we care about the CW data, and this 0 means modulation frequency is 0. I can plot it like this, very nice. And if we can calculate the Jacobian, is again this one line. Pay attention that these are just methods of this object, right? And we can also take a look at this banana, very nice. This is some preliminary benchmarking. Well, it changes from system to system, of course. So don't take these numbers too literally, but here's a little comparison between our new Python version with the updated solver and this current MATLAB version. So there's about 25 to 40% speed up. And we also have a micro version using the same algorithm, but with reduced functionality. It's got only one file. The idea is that it can be easily integrated into other packages. One of them is Cedalion. That's developed by Alexander von Luehmann's group at TU Berlin. Coincidentally, they're going to give a talk on this new Python package in 10 days, so do stay tuned if you're interested. Okay, now that's the end of the presentation. These are my PhD labs. This is Jana Kainerstorfer's lab at CMU. This is us. This is my other advisor's lab, Professor Bulger Grover. And this is my current lab mates, the medical imaging lab at University of Birmingham. And that's a long list of names of people that I worked with. Hopefully didn't miss anyone. And that's it.
Thank you so much. That was really fabulous. Really good to see what research you're doing, and I'm so excited for the future. That's really, really fun what you presented.
You for seeing that?
Yeah, and just want to remind people, if you do have any questions for Dr. Cao, please just put them in the chat if you would like. We do have a question that came in before the beginning of the webinar, and the individual asked, in your opinion, how far do you think AI can go in helping with medical image analysis, potentially using some of these techniques?
Right, that's an interesting question. Very far. Well, I would say that it's a little bit beyond my expertise of research, so my opinion may not be the most accurate, but I will say, yeah, it depends on how you use the AI technologies. There are different ways. One is, of course, then data processing, and think we're already using that in low dose CT technology. Basically, you use AI as the image enhancing technique. That is one of using it. The other thing is that maybe going beyond imaging, or if we talk about imaging in a broader sense, we can collect a lot of data through wearables or just basically multiple devices that are in the clinical setting. And the question is that, can a human really process so much data effectively? Right, and perhaps in that sense, we can use AI as a tool to distill information from this vast amount of data that we have. That would be another way. Well, it's hard to really predict future. I mean, if you look at where we are now versus 10 years ago, there's a huge leap, and the leap is only going to be larger, I would guess. So yeah, at least there are clearly some examples that are happening now that clearly AI is going to be helping the clinicians and researchers, and future, I would say, yeah, it's going to be playing a bigger role.
Thank you. Johan Benarati wanted to thank you for sharing your work. And they did state that their understanding is it requires the whole block to compute the dot spatial prior. And they're asking, would there be a way to compute the dot prior in a more real-time fashion, for example, using a hybrid BCI application?
No, that's a very good question. Yes, your understanding is correct. We do need the entire block to compute DOT spatial prior. That is because we do have very limited SNR in DOT recordings. And is there a way to compute DOT prior in more realistic fashion? Yes, but there's a limited by a few things. Well, if effectively it's limited by the SNR itself, and how do we improve that? Better instrumentation would be, of course, one of them, and then NIRx is here, right? And then another thing is that how can we process the data nicer so we can enhance the SNR, or we can still distill some information out of noisy data. And correlating with the previous question, maybe AI technology can be used here such that even SNR is not so satisfactory, can still do something meaningful, and in that sense we'll be moving towards a more realistic real-time reconstruction of DOT prior. That's what I think.
Awesome. And I think maybe time for one more question that popped in. And this is a little bit more of a basic question that some people might be interested in. I noted that in your 2023 paper in NeuroImage, you had a 24 source, 21 detector montage for your dot configuration. Yes. And there was the question about, in your opinion, is there a minimum number of source detectors or overlapping channels that you need in order to do a dot configuration or reconstruction in general?
Well, that depends on how large the region of interest is. I would say is more related to the density itself rather than the exact numbers. Is there a minimum? Well, theoretically, you can do it with any number, but it's not going to look very good. Of course, I would say the rule of thumb is that if you have this 2 centimeter-ish separation between source and detectors, and you have a reasonable density, you should be able to do something. But it's really a case-by-case thing. It's hard to give a exact number. But there's no maximum that I know.
I mean, that's a fair answer. More is always better, right? All right. Well, again, thank you so much. That was so much great information in there, and just really want to give a thank you for presenting that, and thank all of everybody who's logged in for coming to this webinar, and hope you all have a great day.
Thank you very much for having me. Thank you much for your time.